Thank you, Dame Janet, for very nice introduction. Also, thanks to Stephen for invitation. With my warmest congratulations to the PDB 50 years anniversary. I'm honored to have such an opportunity to present our work and to see so many pioneer crystallographers and structure biologists at this symposium. I didn't realize until yesterday, I checked, my lab has contributed 481 entries in the PDBank. And yeah, apart from protein structures, we have also solved 6 different classes of more than 30 kind of viruses particles, from smaller one like a piconovirus to larger one, herpes virus, even giant one, Afghan swine fever viruses. The title of my talk is Living in the Cell, Understanding SARS-CoV-2 Replication and Transcription from Structures. I hope this published and unpublished result will greatly enhance antiviral drug development. We started coronavirus research since SARS outbreak in 2003. Since then, we have solved most of non-structured proteins of COVID. This experience helped us to give our fast response to COVID-19 pandemics. Within nearly 2 decades of persistence, we have published 51 research papers in this area. Total citation of this work are over 6,000 times. Average citation is over 100 times. SARS-CoV-2 is a causative agent of COVID-19. It shares high sequence homology with SARS. The genome of COVID is the largest one in all RNA viruses and encode 16 non-structural proteins for virus replication and transcription. Those non-structural proteins play key roles in virus replication and transcription inside of a host cell. Some of them relate to RNA and mRNA synthesis, and some of them relate to the host interactions. But those non-structural proteins do not function as individual proteins. They must form an organized molecular machine. We call this protein machine as replication and transcription complexes, RTC. How RTC is assembled and how it regulates the virus life cycle are most key questions to understand SARS-CoV-2 lives in the host cell. Based on our knowledge, SARS-CoV-2 replication transcription involved in several key steps. First, NSP12 RdRp interact with cofactor NSP7 and 8 to form a central RTC, simply called C-RTC. But C-RTC cannot function in real virus life cycle because SARS-CoV-2 genome has many high-ordered RNA structures. This structure must be pre-processed by NSP13, the helicase, into single-strand RNA, and then to be used as a template for RNA synthesis. So in the second step, NSP13 helix must be recruited to C-RTC, form a bigger complex. We call this as elongation RTC, E-RTC. From here, the replication and transcription where we start. Yeah, and for replication, the elongation will go ahead, but for the mRNA transcription, it leads a Cap1 structure at 5' end. This Cap1 structure is synthesized by following 4 steps. First, First, the RNA 5' triphosphate domain RTPase of NSP13 removed one phosphate group from 5' end of pre-mRNA to form diphosphate RNA. Second, leading domain of NSP12 will aid the GMP to remaining diphosphate end. This step produce a cap co-structure called GPPPA. Third, a N7-methyltransferase in NSP14 methylated and the first G to form a cap 0. NSP14 has an exonuclease domain at the N terminal for proofreading. And the final step, and a 2' O-methyltransferase domain in SP16 methylates the second A to form a cap 1 structure. After that, the mRNA will be transferred to the host ribosome for protein translation. Recently, PDBank selected our C-RTC structure as molecular of month. In the introduction, they mentioned one of our work published in 2005, the structure of NSP7 and NSP8 supercomplexes. They introduced this work as first attempt to study assembly of coronavirus RTC. In this work, we determined crystal structure of large hexadecamer complex formed by NSP7 and NSP8 based on these large complexes. Several other works, and we proposed a model for how these large complexes synthesize primer and deliver this primer template pair to RdRp. This paper was published in 2005. I'm very pleased that it is now to be considered as study point to know the assembly of RTC. Benefit from many years consistent studies in this field. We responded to SARS-CoV-2 very quickly. In early January of last year, we got the sequence information from Professor Zhengli Xi, Wuhan Institute of Virology. My Tsinghua group used only 3 weeks, expressed all non-structural proteins to constitute of RTC. And my ShanghaiTech lab, with excellent expertise in cryo-EM. This joint force is greatly accelerated our research. Afterwards, entry the first round protein translation, C-RTC formed by NS12 cofactor and the cofactor NSP7-8. This is a central building block to form other RTCs. NSP12 has a classic RdRp core. It also has a coronavirus featured N-terminal nucleotide transferase domain called nearing domain. Nearing domain is connected to RdRp core by an interface domain. The structure has 7 conserved polymerase motifs, motif A to motif G. In motif C, the catalyzed trial SDD catalyzed the polymerase reaction. And motif A, motif B, and motif F constitute of NTP channel to transfer NTP substrate and nucleotide analogs inhibitors, for example, Remdesivir. After determining the structure of C-RTC, we would like to know mechanism of RNA synthesis Remdesivir inhibition. To answer this question, we determined the structure of C-RTC in complex with template primer RNA pair, as well as Remdesivir. We designed 2 RNA pairs. One is pre-translocation stage, and another one is in post-translocation stage. During formation of C-RTC with RNA, we incubate triphosphate Remdesivir with these complexes. In the structure, we find a very interesting feature. The synthesis of RNA did not immediately stop after incorporation with Remdesivir. An additional nucleotide was linked to the downstream of Remdesivir. So how Remdesivir inhibits RNA synthesis? If we check the structure, we can find that side chain of serine 861 is closer to the robust group of the third nucleotide on the upstream Remdesivir in the nascent RNA. So if RNA synthesis goes ahead and push Remdesivir to this position, the chemical modification on Remdesivir rubbers will clash with the side chain of this serine residue. This would be explanation of inhibition of mechanism of Remdesivir. As I mentioned before, there are many high-order structures in large genome of SARS-CoV-2. This structure must be pre-processed by helicase to single-strand RNA. So the helicase must be assembled to C-RTC to form a bigger complex called E-RTC to initiate RNA synthesis. As a key part of E-RTC, NSP13 belongs to the SF1 helicase superfamily. The crystal structure of NSP13 has solved and reported by my group in 2019. We designed a template primer-RNA pair to form E-RTC. The template RNA has a 5' extension. We assume this extension would recruit NSP13 into E-RTC. This strategy is referenced to the work of Dr. Compl group, Rockefeller. By using this RNA, we successfully determined the structure of E-RTC. To our big surprise, although the RNA has only one template extension, but two helicase molecules were found in RTC. One binds template extension, but the other one does not bind any RNA. Because of high quantity of pri-EM density, we are clearly traced RNA. The double-strand part bound by NSP12 and the two NSP8 non-helix. The template extension is anchored by one helicase. This part binds in a RNA binding channel formed by helicase 1A, 2A, 1B domains. Here is highlight of the extension of 5' template bind to the helicase. Please pay attention to the residues 178 arginine in the 1B domain, which plays key role in our story. To here, we have nailed the structure of RNA elongation. This is a process by an animation. However, here's a key question we still don't know. What and how unwind nascent template product double-strand RNA? We are now working on and try to get an answer. And after here, the 4 steps capping action will continue and add cap1 structure at the 5' end of pre-mRNA. In the first capping step, the RTPase activity of NSP13 remove one phosphate group from 5' end of pre-mRNA. In one of our recent work, we find that 2 E-RTC will form a dimeric structure and use as in trans RTPase polymerase coupling mechanism for the first capping action. We named it as a cap-2, -1 RTC. Here, we can see the moving of this process. This is in trans. Well, in second capping action, chromovirus lead GTase guanylate transferase action to aid GMP to diphosphate end of pre-mRNA. The product GPPA structure is called Compl structure, but the key enzyme of this step is unknown in the past 20 years. In our research, we first discovered that NSP12 leading domain is the enzyme to catalyze this reaction. With the presence of NSP13, NSP12 layering can catalyze the formation of cap co-structure with high efficiency. This is an animation to show this process. Okay, so after the first and the second capping action, let's look at third and the fourth capping actions. So NSP9 is a virus-encoded protein. It was initially known as a single-strand RNA binding protein. In fact, it is an adapter to mediate third and the fourth capping action. After second capping action, NSP9 tightly bind with NSP12 layering and separate cap -2 -1 RTC linked to like two wedges. You can see that. Well, the N-terminal of NSP9 inserted into the catalytic center on NSP12 layering and attenuated GTase activity. The binding of NSP9 in RTC also lead to large movement of ZBD, the zinc binding domain of helicase. In E-RTC, ZBD is on the plane region formed by NSP12 RdRp and NSP8 long helix. After binding with NSP9, ZBD inserted into the minor groove of template primer-RNA. We can view it from the top side. This ZBD is like a bolt to suspend movement of RNA. This means the elongation of RNA is now transferred from the active state to the suspending state. As an adapter, NSP9 will recruit NSP14 into RTC to initiate third capping action. NSP14 is a dual function enzyme. The N7-MT domain for the third capping action in its C-terminus. It also has an exonuclease domain at N-terminus. This exonuclease activities is very crucial for coronavirus life cycle. As we know, coronavirus has very large size genome, but the replication fidelity of RdRp is relatively low. So how ensure this correct replication? Coronavirus has featured proofreadin mechanism. If something sensed mismatched nucleotide in the nascent RNA, NSP14 nuclease will remove this mismatch. We are very, very interested in to know how these two key processes is located in one NSP14. It should be noted that full function of NSP14 required to binding with cofactor NSP10, but NSP10 only binds with NSP14 exonuclease domain and it does not interact with MTase domain. We determined the structure of a RTC composed by NSP9, 10, 14, and the E-RTC. We named this E-RTC as a Cap0 RTC. In the cryo-EM data, we find two kinds of particles. The smaller one is monomeric Cap0 RTC. The larger one is a dimeric form. We determined the birth structure at atomic resolution. In the monomeric Cap0 RTC, the NSP10, NSP14 complex is recruited by NSP9 in the contact with NSP9, NSP12 layering. In the dimeric form, two Cap0 RTC monomeric are related by two-fold axes. NSP13 has tightly contacted with NSP14 metase domain to stabilize the dimer. Here, we are in the first time to know whole architecture of SARS-CoV-2 E-RTC, sp12 leaving sp9, sp10, sp14 related to capping action. They are in here. We named this part as co-transcriptional capping complex, CCC. This is an N7-CCC. The other part are related with RNA synthesis. We named it as elongation complex, EC. In N7-CCC, NSP14 exonuclease domain contact with NSP9 and NSP12, but NSP10 and NSP14 methods are not involved in the formation of caps 0 RTC. The catalytic center of NSP14 methods is on the opposite side of the catalytic center of NSP12 layer. So the 5' end pre-mRNA should follow this pace from second capping action to the third. Okay, here's another animation to show this process. But this pace is one RTC protomer is occupied by NSP13 in another RTC protomer in the cap0 dimer. In this dimer, NSP14 methyls in one protomer tightly bind with NSP12 RdRp thumb and the finger domain. And the NSP13, what is the function of this dimer? After carefully analysis, we know that this dimer is related to proofreading. Proofreading of DNA and RNA. Synthesis is first discovered in the eukaryotic cell with the backtracking mechanism. For the normal synthesis of DNA or RNA, the template move forward in the guided synthesis of nascent chain. But if mismatch in the nascent chain is detected, the template will move back. The lead nascent chain move back together. The 3' end of nascent chain will move outside of polymerase active center and will be processed by some other protein to remove the mismatch. Let's go back to our structure of the cap0 RTC dimer. In one protomer, the catalytic center of NSP14 exonuclease is distant from 3' end of nascent chain. The distance is around 80 angstrom, but it's close to the 3' end of nascent chain in another cap0 RTC protomer. The distance is around 35 angstrom. This indicates us if nascent chain move back 35 angstrom, the 3' end will be reached to the catalytic center of NSP14 exonuclease. A backtracking mechanism for proofreading would be reasonable. But what and how drive this backtracking? Actually, the question is still unknown in the eukaryotic. When we compare the structure of dimeric cap0 RTC with other RTC structure, we found the previously observed large conformation change of 1B domain in the helicase with bind template extension. The 1B domain of this helix has a downward movement around 90 degrees. Such significant conformation change of 1B domain is not observed in coronavirus helix before, but also not reported in all other members of SF helix family. The 90-degree shift of 1B domain leads an important consequence. First, the interaction of helix with template extension has changed. In E-RTC, 1B domain is here. And here, the arginine residues at 178 position anchors tens of nucleotides, but in the RTC dimer, this arginine moves downwards. The distance is just 35 degrees, 35 angstrom. This indicates if 1B domain moves back to its normal position, it will lead to template extension backtracking 35 angstrom. Such movement will drive 3' end of nascent chair backtracking 35 angstrom. Based on this analysis, we propose an entranced backtracking mechanism for proofreading. In the long RNA elongation, helicase pushed template move forward. Once primers sensed mismatched nucleotide, the backtracking mechanism is triggered. 1B domain of the helicase will move downward and then move back its normal position. During this process, 1B domain leads the template extension backtracking 35 angstrom, and 3' end of the nascent chain will also backtracking 35 angstrom. After that, S14 in another C-RTC will be recruited and remove the last mismatched nucleotide. Finally, once mismatch is corrected, RTC will return to the monomeric state and restart normal elongation. This animation shows whole process of entranced proofreading. Mismatched, and then backtracking. Yeah, exonuclease come over. They cut the mismatch back to normal. Okay, finally, we try to elucidate mechanism of the last capping step. NSP16 is the last non-structured protein encoded by SARS-CoV-2 genome. It has 2' O-methyltransferase activity. This activity can methylate the second nucleotide cap co-structure to generate cap1. Its activity also required bonding with NSP10. We reconstitute of ERTC formed by NSP9, 10, 16, and the E-RTC. We name this E-RTC called cap1 RTC and determined this cryo-EM structure. In the cap1 structure, RTC, cap1 RT structure, one NSP10, 16 complex is recruited by NSP9 into RTC. NSP16 phase NSP12 layering in a head-to-head mode. As defined NSP N7-CCC, we named the part formed by NSP9, NSP12 layering, NSP10, NSP16 as 2O-CCC. In 2O-CCC, NSP16 stand upside down at NSP12 layering. It kept that side is also on the opposite side of NSP12 layering, like that in N7-CCC. The pre-mRNA pace goes from backside to front. Let's see the movie. Okay, this is the last methylation. Okay, up to here, we have defined a framework of SARS-CoV-2 replication transcription cycle, determined almost all of the key structures, but we still have many questions need to be answered. For example, what and how unwind template product double-stranded RNA? Second, how to sense mismatch? How to sense the correction of mismatch? Third, how to transfer mature RNA to the host ribosome? And additionally, can other non-structured proteins like SP1, 3, 4, 6 participate in RTC function? How could RTC be localized with a membrane? We hope we could answer this question in the future. Well, this is a 3D model we made, and because of time, I cannot, use too much time. And, the work has done by my colleagues and students from Tsinghua University and ShanghaiTech University. Professor Zhi Rao is a main player. And Dr. Li Ming Yan and Yan Gao, Chuan Wang, and the others also made great contributions. Thanks. This work also done by multiple national, international collaborations. So let's work together to fight against the SARS-CoV-2. Thank you for your attention.
Thank you very much indeed, Professor Rao. That was an amazing romp through this process, which is incredibly complex. My goodness me. But it's wonderful that you managed to tease this apart with all these structures. Absolutely fantastic. So could you tell, there's one question that's been posted, and I guess this is the question that everybody is asking you. How can you use all of this information to develop drugs that will combat the virus? I mean, this is a big question, and I'm sure you think about it all the time.
Yes, thank you, Janet. This is really what we want to do. Of course, I still believe that international collaboration, I still believe that sharing all the information with everybody and scientists, industry people, and we make a joint force to combat this one. Why? This cannot be done by my group. We should join together. So we did that. We are working very, very hard on that. Yeah. Thank you.
Very good. Very good.
Thank you.
So this was all done by crystallography, or were you using EM or? Yeah,
actually, this whole structure being done by cryo-EM rather than crystallographer.
Oh, right.
So because there's an RdRp and SP12, we spent more than 10 years, cannot get crystallized. So yeah, this is why we persistence for last 18 years, 17 years. We have so many people working on the crystallization of ASP12, but we get good sample, but no crystal. So
yes.
Thank you.
But that's why EM is so powerful now, I guess.
That's true. That's true. But crystallography is powerful as well. So we have we have solid foundations for the high resolution structure. We have a good hand for the protein purification.
Indeed, that's very important. Would you like to say anything about vaccine development, or do you think that this isn't appropriate?
I should mention, but because of time, and my group has been involved in China. There's 2 big vaccines and work. We've been participating in that. These 2, market in China, the vaccine. We have been extensively involved in. For example, one of the vaccine, the inactive one, they lost spike. Initially they lost spike. We use cryo-EM and said, well, this cannot work because they no spike. So then we changed the purification and preparation procedure. We get the spike back. So yeah, we've been extensively work on that. So yeah, we are good at on the virus particles. So now we are try hard on this, the new vaccine work. Of course, joined by other expertise. This is a joint force. Yeah.
Thank you very much.
Thank you.
Fantastic work, and thank you for being up rather late. We appreciate it.
I apologize because I've been here, but my colleague used his name, register. Yeah. So that's my fault. Yeah.
No. Well, we apologize for the mix-up. So thank you very much.
Thank you.